The topic that I'm going to present is actually nothing new, but it is intended to raise awareness of the opportunity to conduct high throughput and economical marker-assisted selection for peanut. So there are numerous applications for marker-assisted breeding. These are some of the ones that we are using in our program. Genetic purity of the individuals that are selected as parents before a cross is made, we have found to be extremely useful to establish. So if one has markers that can identify both the male and the female parents and ensure that the individuals that are selected from cultivars or breeding lines that are going to be used as those parents actually contain the markers and their associated traits, then that is very useful. Even though a cultivar is released as a pure inbred line, we know that there is some level of contamination in cultivars, perhaps due to outcrossing. We also use markers to confirm crosses. F1 hybrids are simply putative F1s when they're harvested. Because peanut is difficult to emasculate, we know that it's not always 100%, and it can that percentage can vary from operator to operator depending on the level of experience. And so if one confirms that those F1s are in fact true hybrids, then it will save a lot of downstream time for the breeding program. And of course, ultimately, the use of markers is going to be most applied for marker-assisted selection, using those markers that already have been associated with particular traits. And in peanut, there are quite a few traits now which do have associated markers. One is the high oleic to linoleic acid ratio that's conferred by fatty acid desaturase mutations, resistances to a number of pests and diseases such as nematodes, leaf spots, both early and late, white mold, and tomato spotted wilt virus, and other quality traits such as seed size. So we've been using marker-assisted selection in collaboration with the breeding program of Corley Holbrook for about 12 years now. As I mentioned, to confirm genetic purity of the individuals that are selected as parents, and then also to confirm that the F1 hybrids are actually hybrids. This first attempt at marker-assisted selection was initially quite low throughput, but it did ramp up towards the end of the project, and we now are screening 10 to 15,000 individual seeds per year, and for 2 to 5 markers per seed, and so it's become a very large component of the breeding program. This first selection, however, led to the release of Tifton v. High-OL, which is a nematode resistant line that combined high oleic acid with the nematode resistance. In parallel, we had been also conducting a back-crossing program with the thought that we would release a high OL TIF guard, although Tifton v. High OL was actually released because it was better performing than TIF guard. And the reason I mention this is because in back-crossing, it was possible for us to use the heterozygous individuals to make the next back-cross, because the two alleles would segregate 1-to-1 in that back cross, and therefore we could recover even the recessive mutants in the next generation. So breeding for high oleic acid in peanut requires mutations in 2 genes. As I think all of you know by now, peanut is an allotetraploid with 2 subgenomes, and this chromosome spread on the left, one can see the A subgenome in orange and the B subgenome in green. And so within each of these subgenomes there is a FAD2 gene, fatty acid desaturase 2 gene. The gene in the A subgenome compared with the B subgenome has single nucleotide polymorphisms that can distinguish those two subgenomes, as can be seen in these two positions. Within the subgenome, there are mutations that now have altered functionally each of those genes. In the case of the FAD2A, the mutation is a single nucleotide substitution that changes an amino acid and thereby renders the protein product non-functional, the enzyme non-functional. The mutation in the B subgenome is an insertion, a single nucleotide insertion, and this insertion causes a frame shift and a premature stop codon, and therefore there is very little of the transcript produced, and what would be translated would not be a functional FAD2 enzyme. And so each of these mutations as single nucleotide polymorphisms can be detected by a variety of SNP assays. These are two types that we are using. One is a competitive allele assay known as KASP. Another is a high-probe melting curve assay. In either case, one can detect the heterozygotes, either these providing an intermediate signal in the KASP assay, or curves where there are two peaks, as can be seen in this melting curve assay. So in the case of the fatty acid desaturase, the SNP markers or the mutations are actually within the gene. And that is actually the ideal marker type. A marker within the gene is unlikely to segregate from the function of that gene. However, most of the markers that we have are simply associated with a gene. So they are linked, and in many cases closely linked according to genetic data, but not within the gene, because we actually don't know what the genes are that are conferring these disease resistances at this point. So the SNP markers for nematode resistance are coming from throughout a region that was introgressed from Ericus cardinacei, a wild relative of peanut. And this introgression is shown here in green in Tifgard. But we do know now that we can recover some recombinants, although recombination is suppressed in this region. We can identify some recombinants in the smaller segment that is about 3.5 megabases in length, actually carries the region that gives the strongest nematode resistance. And so we have designed KASP markers, numerous KASP markers actually, along this introgressed region that can be used in breeding for nematode resistance. And so the KASP assay is a relatively economical assay. We are performing that in-house because we have the equipment and we have the expertise to do it. The cost is less than 70 cents per sample, and it will allow us to identify the fixed homozygous resistant, fixed homozygous susceptible, and the heterozygous individuals, and then decide what to keep for the breeding program. As I mentioned, sometimes we would keep the heterozygotes and use those for backcrossing. In other cases, we may only want to keep the homozygous resistant to then look for other traits by field testing. So if one does not have the instrumentation and the expertise to do the genotyping in-house, this is an excellent alternative. It's an economical platform for peanut genotyping that actually was developed through the high-throughput genotyping effort of the Excellence in Plant Breeding program with the CG centers. And they were using are using the Intertek SNP genotyping service that is based in Sweden, although they have genotyping centers also in India and Australia. We have actually worked with the one in Sweden. The basis of their service is the KASP marker assay. I say it's very economical because the per sample assay cost is around $2 for 10 SNPs. Shipping from the US is fairly expensive, but it still works out to only about 33 cents per sample. And so it's still a bargain to use this Intertek genotyping service. It also does not require that you send DNA. In fact, you can just send tissue samples. These can be leaf samples as shown here, and they provide instructions on exactly how to prepare these leaf samples. It's simply a leaf disc or 2 leaf discs per sample that are dried, either oven dried or lyophilized. Alternatively, and we pioneered the use of these seed samples with Intertek, is the use of seed chips. And the seed chip allows you to spread out your genotyping over a longer period of time during the winter when you're not so busy in the field. It's really difficult to collect leaf samples at the right time from the field, have them genotyped, and then make decisions because a lot is happening at the same time. So the seed chips now have been adopted by a number of breeding programs, including ICRISAT, and it's working very well with the Intertek genotyping service. Of course, with either the leaf or the seed chips, it's necessary to somehow tag the seed or the plant so that the assay can be corresponded with that individual. And all of the samples are arrayed on a 96 well plate with the coordinates, so that that correspondence is quite clear. So a couple of years ago we did a test with Intertek, and the objectives were twofold. One was to compare leaf with seed samples, and then the second was to compare our success with the assay versus Intertek's success. And in fact, Intertek had a higher success rate than we did, and this particular assay with the FAD2B mutant, we actually had about 10% of samples that were not successful. And either leaf or seed tissues worked very well. So now we want to expand the markers that are available through the Intertek platform using mapping results and marker associations for a number of disease traits that we have identified, as well as other labs, particularly for white mold resistance, early and late leaf spots, and tomato spotted wilt virus resistance. And so if you're interested in any particular markers or traits, I invite you to contact me or to contact Dr. Holbrook as we begin to expand the options that are available through Intertek. So in summary, of course, marker discovery is upstream and dependent on the populations and the phenotyping that's conducted with those populations, in addition to the genotyping to discover those markers. But once those marker trait associations have been identified, they can be applied and save a breeding program time and resources, and I think we have adequately demonstrated that. If a program does not have the equipment and skilled personnel to conduct marker-assisted selection in-house, Intertek affords a very economical opportunity for outsourcing the marker assays. And so finally, there's been a lot of partners that have contributed funding towards the marker discovery in particular, and more recently we have a NEFA grant that is funding the expansion of the use of this Intertek platform. Thank you for your attention.